Hello friends, in the last two videos we discussed about generalized transduction and specialized transduction in bacteria. You can refer my videos, the link is given in the description box. In this video we are going to discuss about applications of transduction. Transduction has numerous applications in molecular biology and biotechnology. Let us discuss them one by one. First is genetic mapping. It is useful in genetic mapping. Genetic mapping is the process of establishing relative location of genes on a chromosome. So, location of genes on a chromosome is established by genetic mapping. By analyzing the frequency of transduction events, the order and distance between genetic markers can be deduced. So, order and distance between genetic markers are deduced by analyzing frequency of transduction. This provides insight into bacterial genome organization. So, by genetic mapping, we can find the organization of bacterial genome. Second is gene transfer and genetic engineering. Transduction can be used for gene transfer and genetic engineering. Specific genes of interest, which are foreign genes, can be introduced into recipient bacteria. Hence, desired features are transferred or gene function is studied. So, it helps to transfer desired features and to study gene function. This is beneficial for bacteria that cannot be transformed using other methods. Example, some strains which lack competence. Hence, transformation cannot be used to transform this type of bacteria. Small size of fragment is transduced as phage genome is of small size. Hence, small fragment of donor bacterial DNA fragment or part of bacterial DNA and viral DNA can be transduced or transferred. Third is gene therapy. Transduction has the capacity to be used in gene therapy in future. Gene therapy is a science having the aim to correct genetic problems by delivering functional genes to target cells. Research is going on to use viral vectors to deliver therapeutic genes which helps to cure illness into specific cells or tissues using transduction. Fourth is antibiotic research. Transduction can be used to transfer genes from resistant bacteria to susceptible ones. By understanding the distribution of antibiotic resistance genes among bacteria, this allows to study the mechanism, how resistance genes are acquired, as well as the dynamics, how it is spread in microbial communities. Antibiotic resistance leads to bacterial survival and phage population growth. So, antibiotic resistance helps bacteria to survive. And since bacteria survives, hence population of phage also grows. Fifth is bacterial evolution and horizontal gene transfer. Transduction promotes bacterial evolution. As advantageous features present in the genetic material is transferred between the bacterial species or strains. It helps in study of bacterial pathogen evolution, virulence factor acquisition and antibiotic resistance development. Sixth is phage therapy. Phage therapy is using bacteriophages to combat bacterial illness. These phages are used to transfer genes or genetic components that provide antibacterial qualities to pathogenic bacteria in order to kill the target pathogen. If we talk about generalized transduction, it is used to study linkage information, gene mapping, comparing genomes of two different bacteria, mutagenesis, etc. This is because in generalized transduction, random fragment of bacterial DNA is transferred from donor bacteria to recipient bacteria, and in specialized transduction is used to isolate and insert genes of choice in animals and plant cells to modify the genetic constituents and get desired characteristics. specialized transduction, specific genes are transferred from donor bacteria to recipient bacteria. So seeing these applications, we can conclude that transduction can be used in various fields like genetic research, genetic engineering, medical fields, and also in studying the dynamics of bacterial populations. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.